You could use the handheld mic if you're switching back and forth.
Oh, okay. This one?
So you can use this one if they're both on.
Okay.
I think I'm going to sit in there.
Okay. Okay, thanks for having us here today. We are the University of Michigan. It's great to be here at what equates to the big kid science fair that is iGEM. The U of M team comes from a varying background, wide varieties of interests, and as such, we pursued a wide variety of projects. As I'm sure some of you can relate to, when you pursue a wide variety of projects, you end up completing not a lot of them. So instead of presenting a single project to you today, we're going to present a series of tools for construction of any synthetic project. Anything from software to construction to fine-tuning. So without further ado, I'm going to hand the floor over to Paul, and he's going to talk to you about a program he's developed for developmental circuit.
Thanks. So upon joining the synthetic biology team, I quickly became familiar with our pipeline to move from initial idea to final product. So I imagine most of you follow a process that is similar to our own. We begin development with topology design, and we quickly move into modeling, whether that be stochastic or deterministic, to get some idea of how our system behaves. Once those two steps have been completed, we move into fabrication, which includes parts determination, such as trying to find parts that will work in our system and integrating those into our system. For the genes, we must synthesize what primers are we going to need to do so, and of course, we run into some problems along the way. So I found myself fascinated with this process as a whole, and been trying to come up with ways to both increase our productivity while decreasing the time it takes to go from initial idea to final product. So this led to the creation of a program known as Plasmene, and the focus of Plasmene is rapid computer-aided biological network design. Essentially what I'm trying to create is an IDE, or integrated development environment, for synthetic biology. For those of you unfamiliar with the term, an integrated development environment is a software package that provides the user with a central place to both store and manipulate information on a given project. So to introduce you to this idea, I've included a screenshot of an IDE that is already written that is used in software development. And this program is known as Eclipse. And to the programmers out there who may be aware that Eclipse is generally used in Java software development, I'd like to clarify that my program is not written in Java, but is actually written in C#. So if you take a look at the screenshot, you can see there's an incredible amount of information available to the user at any given time. You can see the projects that are currently being worked upon, type and member information that support those projects, and the source code, which is essentially the meat of the project. And I consider analogous to topologies in synthetic biology. So with this picture in mind, I've created a screenshot of what I hope my program to look like in the coming months. So you can see here that there's also an incredible amount of information available to the user at any given time. So if we look carefully, we can see there in the center there are two windows. There's a topology design window where we can quickly create and manipulate topologies. And next to that on the right is a modeling result window where we can get instant feedback on any design we have created. If we continue moving to the right, up in the top corner we have a project explorer, which really contains all the information in the project, such as parts that we are looking to use, topologies we've designed, and modeling results on those topologies. Moving down, we move to the properties pane, which contains information on whatever object is selected in the topology design window. So if we look closely, we can see in the topology design window that the reaction arrow is highlighted. And as such, there's reaction information in the property pane. So we can see what are the reactions and products and get information on those, the kinetic law that tells us how the reaction proceeds, and really any other information that would pertain to the objects that are currently selected. If we move up to the top, where the part palette, we can see a listing of all of the parts that we can drag down into the topology design window. Currently, we're in the generics tab, meaning all of the parts that are shown are not tied to any specific biological entity. But if we look, we can see that there are a host of other tabs, such as genes, mRNA, protein, etc. And if we were to click on those, it would show us a filtered list of all the parts in the Project Explorer that correspond to those sections. And then down at the bottom, we have a miscellaneous panel, which contains information such as the jobs the program is currently working on, host and Plasmene information, and other properties. And to give you an idea of where things currently stand, here's a screenshot of the proof of concept version I developed a few months ago. You can see we still have a topology design window here, and we also have the properties panel on the right, which allows us to manipulate objects in that topology design window. And if you look up at the top, you can see there's an export to SBML feature highlighted. And you may be wondering, why is that important? Well, SBML stands for System Biology Markup Language, and is a generally accepted format for exchanging information from one biological software tool to another. So what we can do is export this topology to SBML, and that can be imported into another program, such as the JARNAC simulator or Mathematica. So if you were to import it into the JARNAC simulator here, we can get time course information on the topology we've created. So now to go a little bit into the technical details, work on version 2 has begun, and as I mentioned earlier, is written in C#. The user interface is based on a variation of the model view presenter design pattern, and is currently composed of 2 independent sub-projects, a diagramming component and a biological class library. The communications project is also underway and includes libraries that will help ease integration with websites, web services, and databases. So if you are interested, the proof of concept version is available for download from Michigan's team page on the iGEM wiki. And if you'd like to learn more, you can reach me via email, or please seek me out during the poster session. And now we'll make the transition from software to wetware, and Steve will take over the presentation.
I'm going to talk to you today a little bit about a system of deliverable nested landing pads built for the chromosomal insertion. For those of you unfamiliar with a landing pad is basically anywhere on a genome where you can insert a piece of foreign DNA. For our purposes, we use target genes such as, or non-essential target genes, such as MALQ, MALP, ARAB, and FUTK for our specific targets. Before I get into the whole design of the landing pad, however, I'd like to talk real quickly about the advantages and why you'd want to use one. The advantage of using a landing pad on the genome versus a chromosomal build is, one, you can limit noise. We've shown in the past, and we'll show later in this presentation, how much you can limit noise of the system, switching it over to the chromosome. You also increase genetic stability. Our system in particular also allows a very simple phenotypic and genotypic screening and confirmation of your insert, as well as a user-friendly stepwise construction. In order to build our landing pad, we started out designing a very simple polylinker inserted into a modified PBR322 plasmid. We then added the first 90 base pairs of our target gene and the last 90 base pairs of our target gene, and an antibiotics resistance gene flanked between the two ends. With the initial backbone and framework complete, we can linearize the plasmid and cross it into a RecD mutant cell, and it'll cross into the genome by a homologous recombination. We use a RecD mutant cell so that the linearized DNA isn't immediately targeted for destruction, and you get a much higher yield. Once the first construct has been crossed into the genome, you can then cross a second construct in by targeting your first antibiotic gene and using a second antibiotic resistance on your second insert. This is one of the most unique features of this particular landing pad. The idea that you can nest or recursively land into one landing pad from another is a new concept, as well as the fact that with each cross, you have a set of antibiotic switching. So you aren't constantly building up more numbers of antibiotic resistances. You're instead switching from one to another, and it leaves you open to do more. Another sort of unique feature to our construct is something that has been proposed and utilized by Andy and others, is the idea of a barcode. And the fact that you can put our particular construct has 21 base pairs at either end that can be used as sequencing primers. So you can check the fidelity of your insert after crossing down to the chromosome. Our initial construction allowed for a custom construct of a single system. That is to say, you could match a custom primer, gene, and terminator as you saw fit. We also worked in a couple of extra restriction sites so that you could adjust and work with constructs that you already had or maybe were already working on. As we've gone along and developed, we've seen a need for much more work with the biobricks, and so we've sort of reverse engineered a patch to make our construct biobrick compatible. What we've done is we've inserted an XBOND SPI site in the middle so that you can build up biobrick constructs in a normal biobricks background and then recursively and then insert into our landing pad and cross onto the chromosome. Thus far, we have completed and characterized one of these landing pads and have 4 more constructed and are in the process of characterizing. That's our little bit on construct. We're going to move on to 2 more sections about how to sort of tune genetic constructs that you've already built. And for that, I'm going to pass the floor to Brian.
I'd like to speak to you today about the ability that we have as fabricators and modelers to adjust the kinetic order of a system. This is a highly studied parameter which has major impact on the behavior of the system. It was studied primarily back in the 70s. But before I get ahead of myself, first let me introduce what it is that, what kinetic order is. So if we consider a Hill equation here with variables y and x, where let's say y is like mRNA and x would be like IPTG. So in a system of, let's repressed by LAC-I, activated by IPTG, we find that this parameter n is it's a Hill coefficient, which also it basically describes a kinetic order. Another word another kind of synonym for that is cooperativity. But anyway, so we find that as we increase the kinetic order, that the behavior of the system becomes increasingly nonlinear. I'll do it one more time just so you can see again. Ooh, look, go. And you see, notice how it's more switch-like as the kinetic order gets higher. But so you may be wondering, why would we care about kinetic order? Why would we want to increase this value? And there's a number of reasons, but for due to time constraints, I'll only mention a couple. One of them is, as I mentioned, there's more switch-like behaviors. So, for instance, let's say in the work done by Ron Weiss on the biological computers, more switch-like behavior is desired. Another reason, of course, is that, as I mentioned before, is they did a lot of studying back in the 70s, increases oscillations. So one such oscillator that we've considered in the lab, which is basically just a simple negative feedback loop here with LAC-I basically repressing itself, otherwise known as the Goodwin's oscillator. It takes a delicate balance of parameters to induce oscillations, as we see in this right graph here. Now, as I mentioned before, by increasing the kinetic order of the system, you can kind of increase oscillations. And that's what we find in these two graphs. The first graph here basically shows, these are the same exact system. The only difference between the two of them is the presence of sinks and the lack thereof. And we see in the first system kind of damps to a steady state, whereas in the second one, there's fully sustained oscillations, which is the desired behavior. And so that's what we like to see. Now, I keep talking about sinks, but I haven't really described much of the physical background behind it. And while I'll leave most of that to Aaron, just a brief overview is that on the different places in the chromosome or the plasmid, you basically put in perfect operators, which kind of serve to sequester away the repressor, and we find that this increases kinetic order. But like I mentioned, he'll describe that for the most part. So this is basically the model that I used in exploring kinetic order and how we can vary that with sinks. And once again, since there's kind of a time constraint, I won't go into it too into depth. I'll kind of gloss over it. But if you wish to find me later during the poster presentation, I'm more than happy to share it with you. But so the results that we get from the model, when we run it through with various parameters and see how increasing the number of sinks affects the system, is exactly what we would expect. So in this first graph, we see that, even though the colors are kind of very light and it's hard to see, I'm sorry, we see that as we increase the number of sinks, basically the system becomes progressively more nonlinear. And these graphs basically are showing more like the number of sinks increasing as the curves go this way. And so it becomes more nonlinear as we increase the number of sinks, as we'd expect. This graph on the right is basically showing that graph if you were to look onto it that way. And likewise, we see that the plane becomes progressively nonlinear, and the kinetic order is increasing, which is exactly what we would expect. And this is what we desire. So, but obviously, you can't just model something. You need to have experimental data to back that up. So, before I go any further, I'm going to pass it over to Aaron, who's going to explain to you what the physical basis behind it, and also some of the experimental results that we got while exploring kinetic order.
All right, thank you, Brian. So as he mentioned, increasing kinetic order can be very important for a multitude of reasons. Now, this raises a couple of questions. Obviously, what can we do? What sorts of mechanisms can we use to raise this kinetic order? And then how do we construct those? And as Brian mentioned, we use a symmetric 20 base pair segment of DNA called an operator perfect, which we just refer to casually as sinks. All right. We originally had started working with some plasmids and checking the activity with seeing how just transforming this segment of DNA into plasmids and into cells with LAC-Y mutant. Wanted to see the expression of LAC-Z in relation to the concentration of IPTG. So what happened was we had a graph basically that showed merely increased basal level of expression, where as we increase the copy number, we saw the basal level just increase. Instead of seeing an increase in kinetic order and a steeper curve as we saw in Brian's models, all we saw was that the graph basically just started at a higher point. We started with PA-CYC 184, which is a fairly low number copy plasmid, which about 1 to 8 copy number. And then we tried as high as PUC 18, which is much, much higher order of magnitude. And it was more like a straight line across. Instead of having any kinetic order, it was practically flat line. We moved on then to sinks on the chromosome. And when we started, we had basically 3 landing pads available to us. One in the RBS, one in GLIN-K, and one in TRIP regions. So the red bars show up here where we inserted the sinks onto the chromosome. And those were constructed using just standard methods of using transduction and P1-Vir and using landing pads. Now the results of that, instead of showing just a raised basal, actually showed more like the models that Brian showed earlier. It showed a gradual increase in this kinetic order. Nothing significant, nothing breathtaking. That is until we saw one strain that we constructed with 4 sinks, which showed dramatic increase in the kinetic order right here. Now this strain was created with, as you can see, 2 sinks in the RBS region, 1 in GLIN-K and 1 in TRIP. Now, you may ask yourself now, what's so special about this strain? What makes it so different from the other strains? And that is a very excellent question. We're currently doing much more work on this. This is a work in progress, obviously. And we have many more landing pads, as Steve was talking about, available to us right now. Some of our observations were that when we went to sequence, the TRIP region was not very clear. The sequencing data did not come back fully analyzed. So we weren't totally sure that these higher number of strains, these constructs, were actually of 4 or 5 sinks. So it's possible that all of these curves right here represent 0 to 3 sinks, where this much steeper curve represents 4 sinks, which has very important implications if it is true, which could mean that this is, with a very, very slight increase in number of sinks, you have a dramatic increase in kinetic order. Now, some of the work that we're doing to rebuild these strains include rebuilding them with different landing pads, with the same landing pads constructed in different orders. We're trying to just try and recreate this and see if we get the exact same results. Also, there's a couple other issues of proximity and location on the chromosome itself. We saw that there's a lot of space on this chromosome. We basically only worked with landing pads on the first half. And if it is possible that sinks on the first half might be a greater effect as replication starts at the beginning and works its way forward, you might have a much more dramatic increase with sinks on the front end of the chromosome. Also, issues of proximity as well. How close the sinks are to one another might play a great factor. These are other questions that we're looking at. Also, the other question of other sinks that have maybe weaker binding affinity, such as O1, O2, etc. So, as Brian and others have said, this could be very, very significant for a variety of reasons for tuning the kinetic order. And I'd just basically like to conclude saying that this was a team effort of a number of people, and we'd just like to thank all the people involved on the team that couldn't be here to present with us, and especially to all of our advisors who gave us great amount of guidance and assistance throughout the process. and go blue.
Were there any questions?
Sorry.
That was the right question.
None? Good. Okay.
Okay.
Sorry, could you please pass around the microphone?
And repeat the question. So go ahead.
On one slide where you had the special result there, which you think was maybe due to forcings and the others were due to 0 to 3. Is the order related to the like the slope of the curve, like at the inflection point, or the change from increase to decrease there?
So the question is basically whether the increase is based on the slope.
Is the order of the reaction due to the slope of the curve there?
This, so is the order of the reaction related to the
To the slope of the curve at the inflection point. The curve starts going up and then it starts going down.
right, this region right here versus say this region right here? Is that? Yeah. So the question is basically is the kinetic order basically is related to the slope of this line in this region. However, it's a little unfair because this is a semi-log plot. It's very hard to just visualize exactly on this, and we don't have any equations that we've modeled that state the slope exactly.
It looks like that other dark curve, I guess it's a blue one in the middle there, and it's pretty much the same slope there. So it's just delayed in reaction. So I still don't understand what the graph is telling me, I guess.
So this is another possibility that just the curve is perhaps not significantly different is what you're noticing, that it's just a increase in time. Yeah, this is another aspect that we need to investigate further that we're still investigating.
You're focusing our attention on the apparent difference in the gain in the transfer function. But the much more apparent difference is the switch, point. Where's the repressor coming from? What's the source of it in the cells? Do you know when you're putting these landing pads in that you're not disrupting the abundance via its expression of the repressor? Good question.
So the question is basically asking whether or not where the repressor is coming from, and if we by inserting these landing pads, whether we're sort of interrupting or altering the repression in some way.
Or its level of expression of the repressor.
Right. I don't believe that it's being affected at all. I think this is purely. I mean, it's not related to.
Is there a picture of the chromosome at all, or?
Right. It's the whole lack.
Because it's being transcribed from like 10 minutes, so it's like next to the Glen K, but I don't think.
It's closer. Yeah. It's the whole lac operand is closer to the Glen K in this region here, but not very close to where the sinks were put in. It's right between the RBS and Glen K, but probably a little bit closer.
So then how do you account for the dramatic shift in the switch point in the transfer function?
How do we account for the dramatic shift in the transfer function? Well, that's pretty much what we're investigating, and perhaps if we're assuming that these the lac-I tetramers are being sort of relayed pretty much freely across the chromosome, and that we're pretty much investigating whether the position and proximity is of great importance or not, basically. That's another good question. Yes?
A couple of questions. What you mentioned, the sequence analysis was inconclusive. What happened?
The question was about the sequencing results and the inconclusivity. It was basically a time-limiting factor that we just weren't able to reproduce the sequencing results soon enough before presenting them.
My second question, how much of this was done with the landing pads?
and how much was done with the landing pads. These were not done actually with the landing pads that Steve was presenting. These were done with previous landing pads. So we're in the process of
basically came to fruition, just recently, like the actual physical, landing pad. So
sort of all these things working together. Do we have time for
yeah,
a couple more? Yes?
You commented that when you had these things on the plasmid, it didn't work out, and the chromosome
Right.
did. Can you comment more on that, on why you think that happened?
Here's the plasmid. Now, there's a couple of different. Oh, I'm sorry. So we were wondering. I didn't have my slide. It was a little bit out of order. I wasn't aware. The question is basically why was there such a difference basically with the plasmid versus the sinks on the chromosome, and if we have any idea why. We have a couple guesses that the repressor, the Lackite tetramers, are being basically on there. There's a few possibilities. One of which is that the plasmid is somehow acting differently, is perhaps just more DNA that's just non-specific binding, and it's not totally clear.
Something else that we believe, what it is. Obviously, as you can imagine, there's sort of like a proximity effect. So the perfect operators are sequestering the protein, and if you can imagine, let's say the protein's being made here, and like that, it's it's in the other room. It's not going to be that effective. Whereas on the chromosome, it's approximately closer. And also, like if you consider like the leading theory of how proteins sort of bind to the DNA, it's believed that rather than like sort of floating around and then binding to the site, rather it kind of crawls along the DNA. So more of the protein has basically a better chance of being on like the DNA where it's produced rather than the plasmid, and so its chances of finding the site there as opposed to somewhere else is great. It's much greater. So that's kind of like the leading theory right now.
How do you feel about modeling? It's a nice talk because you started off with the beautiful theoretical prediction and then you might not do biological analysis.
I would call it beautiful, but thank you.
It's nice seeing those two things side by side. You normally only see one in one talk and then you see the other one the next talk. So is there anything you do differently in the simulation now, or what kind of things you think of the simulators in these long run?
You're saying, so you mean like going back and retroactively changing it based on?
I'm just saying, just in the future, you
Oh.
don't want to design these things and then build them, but what other things are you going to need in a design tool?
Okay, so the question was, what other things are we going to need in the design tool?
What's Plasmene 5.0?
Oh, Plasmene. Do you want to? Yeah.
Yeah, so you're curious as to how Plasmene will be effective in the sinks, in modeling those?
You have to solve the biological problem. What kind of tools do you think you're going to need in a real design tool in the future to build something?
Okay, so what are kind of the features for the next version that I hope to?
Features that, if these are going to be real problems, like where you put something affects how
Yeah.
it works, what's a real editor going to have to look like?
Oh, okay.
I see what you're saying. Yeah.
Yeah, so right now, I guess if we go ahead and look at so I guess if we consider this screenshot, there's not a whole lot else going on behind the scenes at this point. There's an entire biological class library that is functioning kind of on the back end that is in heavy development right now. And I imagine kind of modeling that we could have objects that represent kind of the chromosome of the bacteria. And if you know what bacteria you're using, then you kind of have a little bit more data to speculate with. And so you can tell if this gets integrated with a landing pad idea and you know the sequence of the chromosome that you're using, the program can tell you, can go to EcoPsych or RegulonDB and download the sequence and tell you, well, you're going to insert into the GlimK landing pad. So here's exactly where you're going to be. And then I'm not sure if people have done work on trying to estimate how the DNA, based on just the sequence alone, where you're going to be. But I mean, just by sequence alone, I imagine you could get a pretty good indication of where you're going to stand.
Something like I was kind of trying to understand what you're saying, and something else that in terms of going back and kind of modeling the results and accounting for that, one kind of project that we have in the future, kind of pushing it back because of so the idea basically is that we can hopefully use partial differential equations to model the proximity effect of where the sink is, on the chromosome. Like I said, we're kind of pushing it back because it's a scary thought, but no.
Experimental data on top of the simulation and then saying, okay, there's a
Yeah, definitely, yeah.
Got to explain it.
Yeah, that's kind of like the next step from now, but it's good. Is that it, or? Okay.
I think we're out of time. Thank you very much.